this morning on the Arabidopsis Research Roundup, we're joined by Dr. Martin Howard from the John Innes Centre, and we're going to be talking about a paper which has just been published in PNAS, where he is the co-lead author with Caroline Dean. And the paper's title is Quantitative Regulation of FLC via Coordinated Transcriptional Initiation and Elongation. So, good morning, Martin.
Good morning. Hi.
So it looks to me as if you're looking at a real fundamental issue here in eukaryotic transcription, but through the lens of using FLC. Can you give us a brief description of this paper, please?
That's completely the idea. This obviously the gene we're looking at is FLC, the gene Caroline and also myself studying for a number of years. But the conclusions are hopefully pretty general. So we're using it as a vehicle, really. Although also we want to understand something about how FLC is specifically regulated and how that influences flowering time. But what we're interested in here really is something called the autonomous pathway, which is a pathway that is regulating the level of expression of FLC. And unlike some of the other pathways that regulate FLC, this isn't really epigenetic. It's something that sets up the appropriate level of FLC transcription prior to exposure to cold. There's, of course, another aspect of FLC regulation, which is epigenetic and how it's repressed by prolonged cold. And we're not really talking about that in this paper, but there are some interesting elements about how the two pathways talk to each other that are probably matters for the future. Right. So we're trying to understand how FLC is repressed before cold, non-epigenetically. And what we found, naively, what does one think when a gene is repressed? Well, it transcribes less, so you expect the initiation of polymerase to be reduced. But what we found was that wasn't the only thing that happened. Of course, when the gene is repressed, you do make less FLC transcript. But also what we found, although somewhat indirectly, was that the polymerases moved much more slowly when it was being repressed. So if you're in a state where FLC is repressed, you don't initiate very often and you transcribe slowly. Whereas if you're in an active state, you initiate more often and you transcribe much faster. And that explains some mysteries of some of the experimental results that we found, where we only found sort of twofold differences in the levels of POL2 over the gene, even though we could see factors of 25 difference in the amount of RNA that was produced. Okay. And the fact that we're only seeing a twofold change in POL2 was to do with the fact that the levels of POL2 aren't just controlled by how often you initiate, they're also controlled by how fast the polymerases move. And although in the high expression state you were making 25-fold more RNA, the polymerases were also moving a factor of 10 faster, we believe. And therefore that means you only see a roughly twofold change in the level of the polymerase. So the other, I guess, important thing about this paper was, those results were important, but also the way in which we got it. Yeah. So it was in these systems, it's very difficult to directly measure elongation rates of polymerases.
Yeah, that was going to be my question. How do you actually measure that?
How do you measure it exactly? Yeah. Well, the ideal really would be do a single molecule experiment where you just directly image polymerases going along the gene, but that's not possible in this system. And the predominant way which has been done elsewhere are techniques like Grow-seq, which rely on releasing inhibition of transcription and then measuring how POL2 moves down the gene. But again, that's very difficult to do in Arabidopsis because you can't get rid of the inhibitor efficiently in a whole organism setting. Right. And we tried various other things like MS2, so that we could do live imaging of the RNA, but that didn't work either. So what we were left with is to do a somewhat more indirect measurement. What we could measure were levels of intronic RNA spatially resolved along the gene. Okay. And it turned out, when we did some mathematical analysis of that, was that those profiles contain information about how fast the polymerases are moving. Okay. Basically because if you're thinking about an intron, the polymerase has already takes time also to transcribe over the intron, and the DNA towards the 5' end is also transcribed earlier than towards the 3' end. Mm-hmm. So the RNA lives, the intronic RNA from the 5' end lives longer than the stuff at the 3' end before it gets spliced out. Mm-hmm. And the fact that there's a difference in lifetime there that actually shows up when you actually measure the levels of the intronic RNA.
Okay.
And it turns out that at the 5' end, it's much more dependent on the elongation rate, velocities of the polymerase, than it is at the 3' end. Okay. And that gives you a rather elaborate profile of intronic levels spatially over the intron. And we measured that, and that accorded with some theoretical predictions, which was nice. And that was our way in to working out what the elongation rates actually were.
I see. Okay. So this technique, obviously, that's kind of the novelty within this paper, but are you looking to use this technique looking at the expression of other different genes aside from FLC?
Well, as ever in Caroline's lab, it's rather focused on FLC.
Yeah, of course.
So, I mean, really, this was focused on understanding FLC, but this could be used to any gene. Anything that has a long intron, for example, where particularly we were looking at the intronic RNA here, and we could look at the intronic RNA that was both bound to the chromatin and not bound to the chromatin, the total intronic RNA, taking into account also the fact that these introns are eventually spliced out and then degraded.
Okay,
so we could also look at those levels as well to give that gave us additional predictive power, and then we could measure those and then test those predictions as well, which also checked out. So it is certainly a general technique which you can use in cases where you can't do a more direct experiment.
Mm-hmm. Okay.
Get at it indirectly through measuring these intronic RNA levels. And that tells you something interesting about the gene. It tells you, at least when you're comparing two different genotypes, which is what we were doing here, it tells you the fold changes in initiation. It tells you the fold changes in elongation.
Mm-hmm. And it's helped, I guess it's helped in this case. FLC does have this unusually long intron, I suppose.
And that's right. So it was a nice candidate because it is so big, so you can really get in there and do the spatial measurements.
Okay. And just finally, so that you found something which relates to the chromatin state as well, which is linked to the initiation.
Yeah, so that's right. So what we discovered was that the high expression states and the low expression states have particular chromatin signatures to them. Okay. So the high expression state correlates with high levels of K36 trimethylation, the low transcribed state with low levels of low levels of K36 trimethylation and high levels of K27 trimethylation.
Right.
And there are also signatures at the level of K4 methylation, which accords with some of the things we know about the autonomous pathway regulation, which encodes RNA binding proteins and K4 demethylases like FLD. So what we think is that the chromatin state of this system is determining both the initiation rate and the elongation rates.
Okay.
And this is also bound up in antisense regulation to do with this antisense RNA called CRULAIR that transcribes in the opposite direction across FLC. And we think that's a fundamental part of what's setting up the level of expression by the autonomous pathway. And it's rather an elaborate mechanism where this is still slightly a hypothesis. We can't substantiate all of it. But we think that the fact that there are, between the highly expressed and highly repressed states, there's not only a change in initiation rates, but there's a change in elongation rates.
Okay.
We think that's important because that change in elongation rate alters the dynamic balance in the antisense transcription between choosing different isoforms, splicing isoforms, depending on whether you do a short antisense transcription and make what's called a proximal transcript, or whether it transcribes all the way across the gene and makes a distal transcript. Okay. Which one of those two is selected may very well depend on the velocity of the polymerases. So the fact that you've not just got a change in the initiation rate, but you've also got a change in the elongation rate, we think is important for that regulation. Otherwise, you can make the argument, well, why does the change in elongation rate matter, right? As long as you start transcribing and you finish transcribing, it doesn't matter all that much how long you take.
Right. Yeah.
You eventually make it if you're in steady state anyway. But this argument makes, well, actually it probably does matter what this elongation rate, because it changes the kinetic coupling inside the system, and that changes which antisense isoform you're making, which then impacts the chromatin environment, which then feeds back into altering the initiation rates and the elongation rates themselves. Okay. So it's quite a complicated system.
Yeah, absolutely.
Feedbacks in it, which is why it's so important to do more quantitative experiments, to do quantitative modeling. This is why you have to do the mathematical modeling in this case to make any sense out of these intronic RNA profiles. Otherwise, it's just some totally uninterpretable profile, and it's just too complicated to say anything about it. You've got to have the model underpinning that to say something useful about it.
I see.
And then simply getting these kind of numbers, what is the fold change in the initiation rate? What are the fold changes in the elongation rate? What's the splicing rate? What's the release rate of the transcript from the gene? These are all numbers that are important if you want to quantitatively understand something, which is what we want to do about FLC, because it's an intrinsically quantitative gene that quantitatively regulates flowering. So you want to know these numbers. And, of course, that's something traditionally molecular biologists haven't been all that interested in.
No, right.
So really getting numbers for these things. But, now it's really starting to matter. We're at the stage where you can do this. You can measure these numbers. And it's important to do so, and we're trying.
Absolutely. Okay, that's great. So thanks very much, Martin. So I would encourage people to have a look at this paper, not only for the fundamental interest. I mean, there's quite a bit of mathematics in it, but don't be scared. Biologists, don't be scared to read the paper, and it really can provide some great insight, I think.
We try to write it in a reasonably friendly manner.
Right, okay, that's good. Okay, excellent. So thank you very much for your time, Martin. Thank you.